Good morning. I'd like to welcome you all and start by extending a really, really warm welcome to those of you that are here in Amsterdam in person, but also to those of you who are joining us online from around the world. So it's good morning, good afternoon, good evening, maybe even nighttime for some of our attendees online. So really, really pleased and excited that you've all joined us today. Those of you who don't know me won't know that I've been in the rare disease arena for over 20 years, but I started my life in lysosomal storage disorders. So MPS is a condition in an area that is very close to my heart, and I've made many friends along the way. So again, really excited to be given the opportunity to moderate this morning's sessions. And since that time, I've witnessed commitment, drive, tenacity and determination from this community, and it's brought us to this meeting today, which I think is going to be a spectacular conversation, a really insightful and educational session where we can think about the challenges around developing treatments for conditions that have got neurocognitive and neuropathic challenges, and that situation means that conducting research to generate the right evidence to get the answers and to satisfy regulators and payers is really, really difficult. So this conversation today is really going to set the scene for a new way of looking at some of that evidence generation. And it's really going to expose how biomarkers such as heparan sulfate could really provide a different way of measuring that disease progression. And as I said, I'm really, really delighted, and thanks to Bob and Kim for inviting me to moderate today. Just in terms of housekeeping, so as I've said already, we have a mixture of attendees, both in person and online. The webinar is being recorded, so you will be able to share the link once it's ready for others to enjoy and learn from, but also for your reference. Please, I encourage all of you, particularly those online, not to take screenshots of the slides. We've not gained permission from the presenters to share them. Please don't reproduce them in any way without permission. We will be securing permission from our presenters to ensure that we're able to do that, and we will let you know. I am really pleased that a majority of all of our speakers are here with us in person today, but we do have a couple of speakers who will be joining us online. Peter Marks and David Whiteman, who unfortunately couldn't make it here in person, but have agreed to join us online. And I believe Peter will be doing this at some unearthly hour of the morning where he is. Questions are absolutely welcome, and I encourage you throughout the presentations to note your questions down. You will see on the tables there are a number of index cards. If you have a question, please note that down. Harriet, who is from the MPS Society, who was here a moment ago, who's got her hand up now, will be collating the questions, and we will try to capture and answer as many of those questions as possible. Those of you that are listening online, please submit the questions through the question and answer panel, and again, we will try to ensure that we capture and answer as many as we can. If we're not able to get to them, we will try and ensure that we find a way to get some of those answers back to you. Just to note, this is not a decision-making event. You know, we're not here to change things today, but we are here to inform. We're here to hear from the experts. We're here to understand and be educated on how biomarkers can be a useful tool in some of the activities we want to take in the MPS arena. But of course, this could be paving the way for a future of biomarkers in other rare conditions as well, which would be fantastic. So, in terms of the agenda, we're going to have the first session that will cover the clinical and patient perspective, and the second session talking about the science this morning. And then in the afternoon, we're going to be talking about the currently accepted and evolving evidence in biomarkers and getting some views from the regulators in terms of how they see them and position themselves around it. And of course, an opportunity to have a final discussion towards the end of the day around this. I would ask you all, please, to put your phones on silent during the day. I do understand that we all have lots of interruptions, but it would be great if we didn't interrupt the conversations and the presentations with phones. And I look forward to having a fantastic meeting this morning. So without further ado, I'd like to welcome on stage Bob Stevens, Chief Exec of the MPS Society, to welcome you all and introduce the day. Thanks, Bob.
Morning, everybody. So, I have several roles, but the most important one is I'm a rare dad. 25 years I've been on this journey. That's my this is the honorariums and that I get from everybody, and the MPS Society is the only registered charity in the UK that looks after and supports people with MPS, Fabry, and other rare conditions. Now, I want to bring everybody into the room quickly. I've got 5 minutes, so we're going to zoom in on what a rare life is. And over 25 years, 2 things are certain: uncertainty and time. We live an uncertain life and we have no time. These are my 2 boys, and they were diagnosed and we had to wait for a treatment. There was a randomized controlled trial, but we just had to continue to wait, and we had to wait for years. And there are consequences. So this is Sam now, and Sam is 23, and I get to do silly things like karate with him, so on and so forth, and he's living a rare life. But he got the treatment at 4 years old. This is Oliver, 7 years old. Time. Oliver lives an entirely different life. The reality with Oliver is that I get up in the morning, I go and see if he's alive. I then carry on with my day. That's what time does. So, we live by this motto, our family, and it's by a great Shakespearean actor called Sylvester Stallone, and it's true of a rare life. It's about how hard you get hit and still keep moving forward. That's why we're here today, to move forward. No more time for action. And that is a quote that I always like. Don't wait for the opportunity, create it. We have the people in the room here to create a different opportunity for our families. Everybody deserves to live a good life. We deserve to have a rare life lived better. So let's make it happen, and let's give the next generation the chance that my eldest son does not have. Thank you.
Thanks so much, Bob, and that's a brilliant introduction to Maurizio and Simon, who are going to introduce this morning.
Thank you. So thank you very much. I'm Maurizio Scarpa, and I'm very honored to be a co-organizer of this meeting together with Simon on the scientific part. And let me thank very much warmly Kim Angel and Bob Stevens for having organized this meeting and all the organizing people here. Because as Sheela was saying, we are starting a conversation. It's a conversation about shortening the time and giving patients opportunity. As all of us here, I mean, in this room and remotely, there are actually attending the best people of this field and people that made really this field. And as someone like me working 40 years in this field, we have witnessed quite a lot of progress from the beginning to now. At the beginning, we as physicians were just diagnosing if we knew the disease, and then we were following the family because we are part of the family up to the end. But since 20 years, actually, we have witnessed an incredible amount of progression in terms of potential therapies. And as you see here, we started with enzyme replacement therapy. Now we are speaking about small molecules and how to reduce the storage of these disorders. And now we are also facing the possibility of gene therapy. All these new approaches are very promising. And there are a lot of clinical trials that are actually showing that the way we have embarked is absolutely the good one. And now I leave to Simon just a little bit of discussion how these approaches are witnessed in the clinical trials and potentially what we have to do further.
Yeah, thank you, Maurizio, and thank you everyone for coming and participating. We feel it's a very urgent meeting. For the reasons here, we have there have been many trials over the last 20 years. Some of them we'll hear about today, very promising data. But there have been trials that have come and gone over the last 20 years. Trials that have showed also some promising results and promising data, and yet which did not result in approved therapies and therapies that our patients can offer. This is about patients with children and young adults with MPS disorders that affect the brain. And despite a vast amount of effort, hundreds of patients, these are rare diseases, as Bob has said, but there are hundreds of patients around the world who have given much of their lives to studies, and yet we have no approved therapies today in the US or Europe or anywhere for these conditions, for any of these families. And is that because we have failed in the science? Is that because we're not smart enough? Well, that may be true, but I think a number of these studies have shown benefit, at least partial benefit, if not more, and yet we haven't designed our trials well enough and our endpoints well enough to actually prove that benefit. And so with all of the many hundreds of millions of euros or pounds or dollars that have been spent, and the hundreds of patients that have invested many years in these studies, we have so far come up with nothing. And that's a very hard position to have to accept. And if we were here because we had not made a difference to any of these children, to any of these brains, then we would have had to accept that the science was not advanced enough and our clinical approaches were not advanced enough. But I'm not sure that's the case. And that's much more challenging and leads us to today and the urgent issue of how we do trials and how we approve therapies and assess therapies. And I think we've seen, I think I wrote a paper some years ago with a number of people saying, no, yeah, this is how you do a trial in neuropathic MPSs. This is the endpoints you should use. I think I was really very wrong. And I think we now understand that actually trying to do a study looking at endpoints like cognition over 2 or 3 years, we thought that would be the answer. But actually those studies have been really hard and have not yielded clear evidence. We've actually got a really poor evidence base. The focus that we've all had, very reasonably, on clinically meaningful endpoints that we can all understand, has led us perhaps away from designing the best trials. Because these diseases that we are talking about today do not fit the model of how we do clinical trials. I think that's the problem. And when it comes to how we assess a drug and approve a drug, then the regulators must make a risk-benefit assessment. We, myself as Maurizio and I as doctors, we must make risk-benefit assessments when we decide whether to offer a therapy to our patients. And families, as Bob and others will say today, also every day make risk-benefit assessments about whether an intervention is the correct intervention or not. And we all do that with imperfect evidence. And we also do that balancing the risks of a procedure or an intervention against the risks of not doing that intervention. And that's what's important, because while there are often uncertainties around the intervention, there's often no uncertainty around the disease, which is a certain neurodegeneration and deterioration. Thank you.
And one message, I want to reiterate what Bob was saying. There is a difference if you start your therapy soon or later. And actually, now we have the technology, also with newborn screening, to detect the patient even before the disease is visible. These disorders are not acute. I mean, all the children are born normally, and then they slowly degraded. So now we have the technology really to change the pattern of the disease. I remember 20 years ago when we made an MPS meeting international in Venice, where for the first time we discussed about the blood-brain barrier, which was really something that we thought not being able to overcome. 20 years after, we have now therapy that are showing that this is feasible. So we are not treating, or we don't want to treat anymore only the somatic changes these disorders are making, but now we are targeting the brain. And this makes the difference, because the brain, you will see today, is actually slowing and slow action to be degraded. And now we have the therapies that can really change the history of the neurological involvement. So what we are here today is actually to set the scene, to see how we progressed to path the future of these therapies, because we have the technology, and probably we have more technology than what we can really understand to have. So what we want to do is to use all the technology to diagnose early the child, to treat the child before the onset of the disorder. We have the technology to recognize how the disease will be in severity, and we have evidences that really we can fight the progression of the disorder, and we can fight not only the disease itself, but the major cause of the disease. So thank you very much for being here, and we are inviting also to the next year 2026 meeting in Florence, where we will discuss this with all other aspects regarding diagnosis and quality of life, because treatment is quality of life, and we don't have any time to wait anymore, because we can do something. As physician, when you can do something, you must do something. So help us and help our children to have a better future. So thank you very much.
Thanks, Simon, and thanks, Maurizio. So that takes us into our first session of the morning. We're going to be talking about the clinical and patient perspective, which is a really great foundation to set the scene for the rest of the day. It gives me great pleasure to invite Fiona Stewart, trustee of the MPS Society, to tell you a little bit more about MPS disorders. Thank you, Fiona.
Thank you, Sheela. I'm very aware that many people in this room are extremely familiar with the clinical aspects of MPS disorders, but my job is to give an overview for the people in the room or on the call who aren't just as familiar as most of us are. So MPS disorders, they're part of the group called Mucopolysaccharidosis. There are many different types of MPS disorder, and they're part of the global group of lysosomal storage disorders. They are all inherited. Most of them are recessive, which means that both parents are carriers, and 1 in 4 children will be affected. But MPS 2 is X-linked, where females can be carriers and males are affected. We do get some female MPS 2s, but they're quite rare. And it's very important to realize that because it takes so long to make a diagnosis, very many families will have already had a second affected child by the time the first child has been diagnosed, which creates a huge burden of care for them. So a quick refresher, a very basic refresher about the lysosome. It's a cellular organelle. Most of us who went to medical school learnt about it and forgot about it and never thought about it again for the rest of our careers. And lysosomes contain a large number of enzymes, and they basically break down large molecules into smaller molecules. And sometimes the way I explain it to families is to think that the lysosome is a bit like the recycling plant of the cell. And if any of those enzymes is missing, then the whole process stops and you start to get a build-up of unwanted materials within the cells. And in MPS disorders, there is a build-up of glycosaminoglycans, GLAGs, in the body. And these GLAGs, including heparan sulfate, are toxic and they damage cells. And it's really important that we all remember that heparan sulfate is not just a marker, it is an active promoter of disease. So diagnosis. Well, these disorders are very rarely diagnosed during pregnancy unless there's been a previous affected child where they have prenatal diagnosis, or there is a very severe complication such as hydrops fatalis when the baby becomes incredibly edematous. Some of the features may be present at birth, so we sometimes see skeletal features such as talipes, club feet. You can sometimes see deformities of the chest wall, or a gibbous, which is a lump on the spine, caused by the fact that the vertebrae aren't properly formed. They can have hernias. You may see corneal clouding. There can be a slightly swollen abdomen due to a large liver. And they sometimes have subtle facial features, which might be recognizable to people who've seen it before, but most pediatricians don't recognize it. So although there may be features at birth, they are very rare. They very rarely, in my experience, lead to an early diagnosis. So what happens in childhood? Well, initially, development often seems normal, but then the children start to get a whole range of problems. So ENT issues are a big thing. The children often get large tonsils and large adenoids, which causes them to snore loudly at night. And that's quite unusual for babies. Babies cry a lot, but they don't often snore loudly. They can get middle ear infections. They can get nasal discharge. So almost all of them end up seeing ENT consultants. They get recurrent chest infections. They can get cardiac problems. They get worsening skeletal problems. So these families are seeing a whole load of different consultants. And back in the day when we had paper records, sometimes the charts were almost as big as the children. They can get a bit of a protuberant abdomen because of the hepatomegaly. They can get hernias. And a really interesting aspect is that some of these children, particularly MPS 3, can get features that are often confused with inflammatory bowel disease, which is it's very problematic for them. And neuronopathic MPS, initially development seems to be fine. And then it starts to slow. They don't gain skills quite as quickly as they had been doing. And sometimes they may be seen by people and assessed as having autism or ADHD. And they do have those features, but quite often no further investigations are done to see what the cause might be. And what's very hard for parents is to see the children starting to lose previously acquired skills. They may also get seizures. They get a lot of behavioural problems, and this causes problems when we're arranging clinical trials. These children, particularly type 3, are very hyperactive. They can be quite aggressive. Their sleep is poor, and they start to get signs of dementia later on in the illness. They lose mobility, and that's very distressing as well. When your child who could walk stops walking, very hard day for families. They lose their swallow, so their ability to eat and drink safely becomes a problem, so they may need to have a PEG tube put in, and they have a reduced life expectancy. And this is a paper with Professor Jones, one of the authors, published in association with the MPS Society some time ago. And what's very important is, even in the 2000s, life expectancy for MPS 3 is in the mid-teens, and that's not what any parent wants to see or think about. So current treatments, well, we can do stem cell transplants for MPS 1. We have enzyme replacement therapy, which, if you've heard, alleviates some of the problems in MPS 2, but it's mostly the somatic problems. And the enzyme does not cross the blood-brain barrier, so cannot address the neurological problems in neuronopathic MPS 2. And there are no treatments currently licensed and on the market for MPS 3. So ongoing clinical trials are showing positive outcomes, but it's quite difficult to demonstrate the required clinical endpoints. So it's very important. Treating neuronopathic MPS disorders, specifically 2 and 3, is still a major and as yet unmet need. So what about clinical trials? Well, they are extremely challenging, given the complex medical problems of these families. And always remember, if you have a child who's eligible for a trial, they have an older sibling who is further down the pathway, who is not eligible for the trial, and who has very complex, severe needs. These are rare diseases, so families either have to travel long distances or maybe relocate. And if you can imagine, trying to relocate, if you have children with these problems, is very difficult. You know, arranging suitable accommodation. As I always say, you can't just put them to stay in the Holiday Inn. You know, that has to be really well thought through. And families commit to this. They invest a huge amount in joining one of these clinical trials. It's very hard as well for families to realise that their child is so advanced that they're not eligible for the trial. They've heard about the trial, they're very enthusiastic, and then you have to say, actually, not for you. And it's also quite challenging to prove the clinical efficacy assessments. And we have to always remember that for families, stabilisation of their child and preventing them from losing the skills that they already had, it's a positive result. If you can stop them deteriorating, that is a positive result for a family and a set of parents. Standard clinical trial endpoints don't always reflect families' real-world experience. And as you will hear more, the deterioration can be quite slow and chronic. So it's very difficult to run a trial for the length of time that would be required to show these problems, and it's unbelievably expensive. So the delays in it all are really frustrating for parents. They want it all to happen now because they're watching the child in front of them losing skills. And to explain why the trial has to go on for a very long time is, as all of us clinicians know, really quite difficult. So that, I hope, is a brief overview of MPS disorders and some of the challenges we're facing in treatment and trials. And I think it's always really important that as clinicians, we have to remember the needs of the family with MPS have to be our major focus. They should be at the forefront of everything that we do. So thank you for your attention.
Thanks so much, Fiona. That was a fantastic outline of the challenges that we're going to be discussing this morning. I'd like to give a warm welcome to Mark Dant, who's the Executive Director at the Ryan Foundation. Mark, welcome. I should have also said that biographies of all of our speakers are in your pack, so if you need to know much more about these fantastic people, then please refer to those biographies.
Thank you. Today you will hear from physicians and scientists who are the top peer-reviewed experts in the world. I am neither. I am Ryan's dad. This photo was taken of Ryan at age 3 in 1991, not long after he was diagnosed with MPS 1. 34 years ago, there was no approved treatment for any MPS disorder. We were told to take Ryan home, and he would probably die within 10 to 15 years. For the next few years, I spent every spare moment in the public library reading about MPS, most of which was written by Dr. Joseph Munzer, who will speak here today. The more I read, the clearer it became treatment would come from the global work and partnerships between the patient, the scientist, the physician, industry, and our regulatory partners. In December of 1997, an enzyme replacement therapy trial for MPS I began in the US. Ryan enrolled. And on February 13th, 1998, received his first infusion of the drug that would put back in his system what he was not able to make himself, clearing the stored substrate that by the age of 10 was already destroying his body. 10 patients enrolled in that trial. The New England Journal of Medicine would later publish that all 10 patients improved. The initial review team at the FDA suggested the sponsor bring the data forward for review without a double-blind, placebo-controlled arm, but instead based on the reduction of the disease-causing biomarker. But before the sponsor could do so, the lead of that FDA team was diagnosed with brain cancer and passed away. The application was reassigned to a second team of reviewers who balked at the idea of an approval based on a decrease in storage of the disease-causing biomarker and called for a second trial with more patients and a double-blind placebo control arm. It is at this moment in history where drug approvals for terminal MPS conditions changed for what has now been more than 27 years. Instead of approvals being based on the reduction in the disease-causing biomarker, all MPS approvals have been based on years-long, double-blind, placebo-controlled trials. This decision in my son's trial delayed the eventual approval another 4 years and nearly bankrupted the original sponsor. The second trial gathered the same data as the first. This time, all 44 patients improved with trial sites in Germany, England, Canada, and 2 in the US. They all collected the data that proved that the drug lowered the disease-causing stored substrate, just exactly as it did the first trial 5 years earlier, and just exactly as it had in the canine models years before the human trial. The double-blind placebo control arm also taught us, the patient community, something quite profound. Some of our children would have to suffer through and sustain irreversible damage in order to satisfy regulatory requirements. Since that first MPS I approval 22 years ago, MPS 2, 4, 6, and 7 all have approved ERT. All of those approvals showed a reduction in the disease-causing biomarker, yet all were forced to have a control arm. Despite 2 decades of data proving the use of substrate reduction as a primary disease activity biomarker in MPS disease, global regulatory authorities have yet to accept the long-term reality that the downstream effect of lowering substrate accumulation in all of these disorders has improved the quality of life for these patients and extended life itself. So the EMA, as we know, has one set of standards. For approval, drugs must be shown to be safe, effective, and of good quality. That's it. There's no other hidden regulation. What's interesting is society would never allow double-blind, placebo-controlled trials in terminal pediatric cancer patients because it would be unethical. Yet we allow them in terminal ultra-rare neurocognitive diseases, and I'm wondering why the difference. Meet Sadie. Sadie has MPS IIIA. At 2, Sadie was cognitively ahead of her neurotypical peers and could say her ABCs, count to 10, and spell her name. At 3, Sadie enrolled in a blood-brain barrier crossing ERT trial, and after only a few weeks, her stutter went away. A month later, before any results were available, the sponsor announced a reorganization to focus on later-stage drug development and a plan to divest the Sanfilippo treatment. But patients were allowed to continue the drug for the 2-year duration of the clinical trial. Results showed the drug passed the blood-brain barrier in all patients. Heparan sulfate levels decreased by 70 to 80% and correlated with stabilized cognitive scores on the Bayley scales. For several months after the trial's completions, Sadie remained stable. But as heparan sulfate started to build back up, she started to regress. Over the last year and a half, Sadie has lost nearly all of her once large vocabulary. She can no longer tell her family she wants her favorite food, ice cream, or watch her favorite show, Peppa Pig.
What's this? And what does this say? What's your name?
Sheela Haywood. Ask me again, I'll tell you the same. My name is Sadie. My name is Sadie.
What's your name? What's your name?
MPS disease does not improve on its own. It is scientifically and medically impossible. The reduction of heparan sulfate, the disease-causing biomarker, took Sadie off the normal timeline of this progressive disease. We, the families, know what is coming. Treatment risk is worth the potential outcome. Improved quality of life, not only for the patient, but for the caregiver as well. The risk is the disease. The ultra-rare disease ecosystem simply not aligned with the regulatory process, and transformational science is being left behind. We cannot continue to expect biotech partners, many of them startups, to keep investing in the extremely small market where our children exist when the current system takes years in clinical trials. Our regulatory system must evolve with the science. In 2006, 19 years ago, the EMA issued this guideline on clinical trials in small populations that defines perfectly a surrogate marker. Quote, to be reasonably likely based on epidemiologic, pathophysiologic, or other evidence to predict benefit. Reasonably likely to predict benefit. Everything these experts will speak to today will point specifically and precisely to that definition. Over the past 30 years, I have attended countless funerals for MPS children who could no longer wait for our regulatory processes to catch up to the science. For my son, the partnership of patient, science, physicians, industry, and the government found his tomorrows before it was too late. In addition to 27 years of weekly IV infusions, Ryan has been receiving off-label intrathecal ERT lumbar punctures for over 13 years, which lowered heparan sulfate levels in his brain and stopped the progressive decline he had been experiencing. Ryan's experience demonstrates quite well even a small change in brain substrate levels changed the course of his disease. At 36, he's the longest treated person in the world with an MPS disease. He graduated college, he's got married, and works full-time for the Munzer MPS Research and Treatment Center at the University of North Carolina. The constant evolution of science is giving him the opportunity to live his life and hopefully be here when better science arrives. The reality is today's science can save this generation of MPS children. The EMA needs to demonstrate the courage to update the regulatory process in Europe to catch up to the science. Tonight, as the words of this day echo through your mind, remember these words. My name is Sadie Ray Haywood. Ask me again, I'll tell you the same. We do not want your sympathy. We do not need your empathy. We need action from you, our EMA partners, and we need it now. Thank you.
Thank you so much, Mark. And that takes me to Dr. Kim Stevens, who's the Executive Director at the MPS Research and Treatment Centre. Thank you, Kim.
Thank you. Hello, my name is Kim Stevens, and I am the Executive Director of the Munzer MPS Research and Treatment Center. But more importantly, I'm the mom of a boy with severe MPS 2. His name is Cole. Those are my disclosures. Cole no longer sings or talks. He lost those abilities. At 5, Cole stopped singing. At 7, his words went away until he spoke only 2 words, help and mommy. At 9, he lost his last word, mommy. He hasn't spoken since. It's been 5 years since I've heard his sweet voice. Cole entered an intrathecal enzyme trial when he was 4 years old. Each month for the past 131 months, Cole has been sedated, and he receives the enzyme that he's missing directly into his cerebral spinal fluid through a lumbar puncture. Though Cole no longer speaks, at 14 he can do things I never imagined that he would be able to do. By 14, most boys with severe MPS are near the end of their life. They're often eating through a feeding tube. They have trachs to allow them to breathe. They're no longer walking, but in wheelchairs. Yet Cole and I often go on 3-mile walks. He's still feeding himself and breathing without assistance. And he completes 50-piece jigsaw puzzles. And he loves to run to the school bus each morning. The dose in the intrathecal trial that Cole was in is widely believed to be inadequate. Yet the heparan sulfate levels in Cole's CSF were lowered because the enzyme that he can't make was replaced. But Cole, after years of taking the same 2-hour standardized cognitive test every 6 months for many years, refused to take it any longer. He would do anything to get out of it. He would act like he was asleep. He would crawl underneath the table. And one time he even picked his chair up and turned it away from the neuropsychologist because he was over it. Like any child, he was bored. Unfortunately, that resulted in Cole having a cognitive score effectively of 0. And he wasn't the only one. So the sponsor, after 10 years of compiling data and not being able to quantify clinical improvement through the cognitive tests, to reach a certain p-value, discontinued the trial. Had heparan sulfate been accepted as a primary disease activity biomarker by the regulatory authorities, objective scientific measures could have been used to reasonably predict the clinical benefit that Cole experienced. The reduction of the store's substrate took Cole off the typical timeline. You saw that in the video. Other boys have not been as fortunate as Cole. They did not receive the treatment in time, and they passed while waiting for the very drug that Cole is on to be approved. It was not approved. And not because the drug didn't work. Cole is evidence that it did work. Otherwise, he would be on this slide with the other boys. Allow me to tell you the story of one of the boys on the slides. This is Kevin. Cole tested for the clinical trial in 2014. It was the most stressful week for our family. Cole passed all the tests to qualify for the clinical trial. And I remember sitting agonizingly waiting for the recovery room on that Friday to see if he would be randomized to drug or to the control group. When the nurse came out and said, "Cole's been randomized to drug," I burst into tears. She gently touched my shoulder and said, "Kim, I don't think you heard me right. He's randomized to drug." I heard her, but what I heard was, "Your child's gonna live." I rushed out of the waiting room to tell my friend Shannon, who was waiting on us, "Cole is on drug." As we hugged and cried, another mom was sitting beside me, and she asked, "Is that for the MPS 2 trial?" Yes, I was very excited. My son has MPS also. "Is your son in the trial?" "No, he's too old to qualify." I went from the highest highs to the lowest lows because I knew I was face to face with a mother whose son was going to die soon. And not because there was not a treatment available, but because he didn't qualify for the trial. He, like the others, would have to wait. Kevin did wait, but the drug was not available in time for him, and he died. Our kids cannot continue to wait. We need better outcome measures. We need heparan sulfate to be recognized as a biomarker of disease. If we can reduce the heparan sulfate, we can stop the leading cause of these diseases. We need your help to get there, so no other mothers and fathers watch your children die as they watch helplessly. You can be the difference in our children's lives. Thank you.
Thank you so much, Kim. Dr. Cara O'Neill is working at the Cura San Filippo Foundation. Thank you, Cara. Did you want the microphone?
Thank you. You've just heard incredible insights from Mark and Kim, and it's hard to hear as a parent who also shares that suffering. But these diseases take an unimaginable toll on our loved ones. And you'll hear throughout the day common threads about the importance of time and the incredible opportunity we have to use a scientifically sound biomarker to bring potential treatments to children today. My name's Cara O'Neill. I'm Chief Science Officer and co-founder of Cure San Filippo Foundation. I'm a pediatrician and, most importantly, mom to Eliza, who lives with San Filippo A. From those many perspectives, I'll share more about the challenges our community has faced and opportunities ahead. As many of you know, San Filippo syndromes are ultra-rare, genetically inherited enzyme deficiencies. The 4 subtypes of San Filippo result from enzymes, inefficient enzymes along the pathway necessary for the proper breakdown of heparan sulfate. These inadequate functional enzymes directly lead to the accumulation of partially digested fragments of heparan sulfate. These are toxic to cells, and particularly toxic to sensitive cells of the nervous system. And because of the key role heparan sulfate and its metabolism play in normal neurodevelopmental processes, you often hear San Filippo being characterized primarily by its behavioral and neurodevelopmental features. However, it's very important to remember that HS accumulates throughout the body and does cause multi-system harm. The neurocognitive impact of HS storage burden has led to significant research focus around this particular domain. When children are born, they're seemingly normal and have a fairly typical early development period in infancy. It's quite rare to detect children during this period. We do not have newborn screening for San Filippo. So unless there is an affected sibling, often children are not diagnosed until the typical age of 4 to 6, after they've begun to exhibit significant neurologic symptoms of disease. At this time, they may have already reached the peak of their developmental skills or even be starting to lose skills. In this simple picture, you can see the impact of time. This is my daughter, Eliza, on the right, who not long after her diagnosis, at age 4, she had been very hyperactive, incredibly talkative, playful. She carried also a diagnosis of autism at that time, but that was really her peak. And Isabel, who was like Eliza at 4, at that time, age 11, had lost all of her independence, including mobility. She needed to be fed through a gastrostomy tube because she couldn't safely swallow. She suffered from terrible dyskinesias, dystonia, and seizures before she died at age 15, which is sadly the average life expectancy. And while there's expected variability from person to person, the disease course is well understood. Because of the intense research focus on neurocognition, we actually have the advantage of leaning on data from many observational studies, providing us with a clear picture of the cognitive trajectory. By 5 to 6 and 8 to 9 years respectively in Sanfilippo A and B, these children are falling off a cliff, so to speak. As someone developing treatments, you might look at this as a positive. Great, we can target that right there. And indeed, this has been the standard approach, where cognition has been the primary efficacy target for Sanfilippo programs over the past decade. But has this approach served us well? Strikingly, there are still no approved therapies for any form of Sanfilippo, nor any CNS-directed therapies for any neuronopathic MPS diseases that you've heard. As you can see depicted in the red bars, we have lost far too many promising therapeutic programs along the way. And despite evidence of biochemical effect and encouraging clinical signs reported by doctors and families, these trials were simply not set up for success. By and large, they were run the average industry time span for a trial, about 2 years, which is far too short to demonstrate significant clinical change on a cognitive endpoint. An endpoint that we have learned is not well suited to a symptomatic population. So we are always left with questions about these trials. How would they have proven effective in the long run? Where would our kids be now? And with the known time course of symptom evolution, we have to ask, is it feasible at all for a small biotech to sustain clinical trials for the decade or so that's needed in order to statistically confirm a clinical endpoint? This graph tells us the answer, no, it's not. Without the ability to access biomarker-based approval, companies have repeatedly shelved our programs, returned them to academia, or gone out of business entirely, struggling to maintain these studies. Let's look back at the timeline again. And remember, treating in infancy offers us optimal chance of preventing brain injury. However, to demonstrate that large magnitude of change on currently available cognitive assessments, we'll have to wait 5 to 6 years in the most aggressive form of Sanfilippo A, longer in type B, and even decades in Sanfilippo C and D, which have a more protracted disease evolution. And in the meanwhile, the whole population declines. Every day that a trial is extended before drug reaches market is another day more brain cells are lost for all of the other children waiting. And while we aim to treat as early as possible, let us not forget about the prevalent population. Again, we don't have newborn screening. The 99% of children living with Sanfilippo who are symptomatic, who are no longer babies. To meet their needs, we have to think beyond cognitive assessments as outcome measures, and instead focus on fit-for-purpose measures that are both feasible and meaningful for this group. So what are those? We conducted caregiver preference research to understand this. What are the important domains even after cognition has been significantly impacted? And families from around the world shared with us what they would like to see from future treatments. They want their children to be able to eat and drink by mouth, to be able to move and walk, to sleep, to not be in pain. These are all very reasonable aims. Clinical trials have not historically focused on these key domains. Thankfully, we are seeing sponsors now utilizing this information looking creatively at their data, and incorporating additional measures into future studies. This is encouraging, because capturing this breadth of benefit will ultimately be critical for allowing broader access to treatment across the lifespan. Time. We're racing time in every part of this disease. Time means progressive brain injury. Time impacts the magnitude and domain in which benefit can be anticipated. Time impacts sponsors' ability to even enter into the space and develop drugs. But time matters most of all to the children and families that bear every single consequence of our action, and more pointedly, our inaction. Keeping this in mind forces us to confront the ethics and appropriateness of withholding potential treatments in these irreversible neurodegenerative disease clinical trials. We see this laid out very plainly when CNS-directed treatment is withheld either through randomization or required lead-in observational phase. This is one child's story, Liv's story, but far too many children's experience. Liv was diagnosed at 2.5 with Sanfilippo B, and just 1 month later, her family was thrilled she had the opportunity to enroll in a clinical trial. This trial, however, required a 1-year observational period before active treatment could begin. She was doing pretty well at the start of the study with a developmental quotient around 80, but this dropped precipitously and was associated with a nearly 10% loss in brain tissue volume on MRI. Liv's mom notes, by the end of the observational study, Liv could no longer follow directions, say words. She stopped playing with her dolls. Her sleep got worse. She could barely pay attention. It still breaks my heart to know the medicine was available, but everyone just watched while the disease stole so much from her. Now, after that, Liv did roll over into the treatment phase of the study, but she will never get back what was lost during that critical period of neurodevelopment. As a community, we understand the rationale behind randomized clinical trials. We get it. It's science. We understand. But we live in the real world, and we have to apply that. We have to fully understand and recognize the unacceptable impact of withholding treatment in these scenarios, especially when we have a strong primary biomarker like CSF-HS that's reasonably likely to predict long-term clinical benefit. Science has already told us what will happen in these scenarios. These children will suffer irreversible brain injury. And knowing that it is not possible to truly rescue them and bring them back to the state they were before, how can we continue to put children in this position and ask families to enroll their children in these types of trials? Thankfully, we have the opportunity to do something different, to use the tools and strategies at hand, like CSF-HS as a biomarker, to look to different outcome measures and design ethical studies so that together we can make a positive change on a global level. I want to leave you with a 1-minute video, and as you listen to the speakers today and benefit from their expertise, I hope you'll always keep at the front of your minds the real children and families whose daily lives depend on our regulatory systems to work urgently for their health in the here and now.
Thank you so much, Cara. Mark and Kim. It's been brilliant to hear the challenges that you're posing to us this morning and for the rest of the day. It brings me great pleasure to invite Professor Scarpa on stage to highlight the role of heparan sulfate as a primary cause of the neurological derangement. Can I please also check with the audience, just a reminder, please don't take photographs of the slides. We have asked you not to. We will circulate the presentations and the slides post this meeting. Thank you.
So thank you very much. These are my disclosures. Being around for a lot of time, I have a lot of collaboration, but no direct interest in the marketing. And we are speaking now on a scientific level regarding what is these disorders, what are these disorders for the brain. This is the image, actually, of a normal brain. And these are the images of the brain of the children affected by neuropathic disorders or neuropathic MPSs. And you have heard from the previous speakers all the effects that this kind of alteration are actually giving to the quality of life and to the life of the children. But what are these children suffering from? You heard from Dr. Stewart the different symptoms of children affected by neuronopathic disease, behavioral problems, hyperactivity, sleep disorders, and all of these are increasing in severity and are leading to a very poor life. But what is this similar to what we really know to the brain? This is actually similar to what is happening when you have a car accident. I mean, this is a direct scene made by the fire workers in Italy of a lethal car accident. Here, unfortunately, there is still the lady who died because he was going straight on a curve. And there is a big discussion regarding how to prevent this lethal accident by probably making more efficient belt seats, or actually to increase the number of airbags is also a possibility. But actually, this accident was done because of the brake problem. This car was not having the brakes working. So at the end, what was it just necessary was to go to the garage and to fix this problem. So this is actually what we really need to do with these children. Because, I mean, the heparan sulfate is indeed present in all the MPSs having neuronopathic disorder. MPS 1 Harler, MPS 2 Hunter, and all the MPS 3 and the MPS 7. And the heparan sulfate is not a biomarker. It is a toxic molecule. And evidences are not only in the brain, but also on other systems. For example, here in 2012, it has been shown that even when you have a bone marrow transplantation, if you have levels of heparan sulfate which are increased, the engraftment of the donor bone marrow is actually impaired. And this is why we treat our patient before bone marrow transplantation with enzyme in order to clear indeed the heparan sulfate in the bone marrow matrix. And there are also other evidences, even older, about the inflammatory and the cause of heparan sulfate in the brain. Like, for example, in 1990, there has been the accumulation of heparan sulfate in neurons and the beta amyloid protein in Alzheimer's disorders and Down syndrome. There has been later the evidences that actually the accumulation of amyloid is present also in MPS brains. It has been shown in 2010 that heparan sulfate mediates the amyloid internalization and cytotoxicity, so it's even an agent for other molecule toxicity. It has been shown in this very nice review in Nature Immunology how heparan sulfate is a mediator of the entire inflammation processes, and the neuroinflammation is bringing also to derangement of other compartments in the cell, not only the lysosome like the mitochondria and other cellular part of the cell. And for this, I made this cartoon showing exactly all the processes that are affected after the initial heparan sulfate accumulation. And you see here that there is mitochondrial strife, autophagy, protein misfolding, aggregating protein deposits, ubiquity in proteasome, and neuroinflammation as a whole. So in the normal brain, the cell like microglia and astrocytes are actually supporting neurons in their function. But if there is a neuroinflammation like in Sanfilippo, there are reactive microglia and astrocytes which are actually contributing to the brain inflammation and to the neurodegeneration. And this is actually what you have in the brain of these children. You have cells that are totally unnatural with ectopic dendritogenesis that are filled up with a lot of lysosomal and other compounds that are really undegraded. So, I mean, the proposed cell death is that there is an enzyme deficiency, there is a storage of heparan sulfate, but this is becoming a secondary cascade of events with impaired autophagy, accumulation of toxic protein, accumulation of mitochondria, and then there is a cellular distress with chronic inflammation, and then at the end, the cell death. This is not a very acute process. We have shown, comparing mice and human brains of MPS II, that actually the process is going from neuroinflammation to glial degeneration to neurodegeneration with a lot of time and in different stages of the disease. This means that indeed the disorder is starting from the white matter to the gray matter, and this is why there is the alteration that you've seen at the very beginning of my presentation. And this is also why, with time, we are showing all these kind of symptoms in different severity. And so we have time to go to prevent this alteration and to prevent this alteration in the brain, which has been shown not only in my cartoon, but also in the direct experience of the previous speaker. So what we have to do indeed is to really fight heparan sulfate. And in conclusion, what we can say that the MPSs are indeed the neuronopathic done due to the heparan sulfate progressive cellular damage, that the heparan sulfate study is also important to understand better the symptom management, because the heparan sulfate elevated concentration is associated to manifestation of the MPSs. By reducing the accumulation of the heparan sulfate, we are preventing and alleviating the symptoms. The accumulation of heparan sulfate is causing irreversible damage with time, not just in a short time. And actively reducing the heparan sulfate, we prevent or we can delay at least the toxicity and the brain damage. So, I mean, if we have a prolonged exposure to heparan sulfate level, there is a development of severe complication, and correcting the heparan sulfate level, we have an increased lifespan and a better quality of life for the patients. And so correcting the biochemical imbalance and therefore the accumulation of heparan sulfate, we can address the fundamental cause of the disease. And correcting the heparan sulfate levels throughout therapy, we can really have an holistic elevation of treatment benefit and outcome of the patients. And for this reason, I hope that you agree with what Emil Kakis was writing, that the heparan sulfate is not only a biomarker, but rather a primary disease activity biomarker. So what at the end we have to say? Fight the heparan sulfate. Thank you very much.
Thanks so much, Maurizio. Again, a really helpful understanding of why we're here and how we can, as you say, fight heparan sulfate. It brings me great pleasure to ask Professor Simon Jones to come on stage to talk about why we need biomarkers in clinical trials. Simon.
Hello, everyone. I'm in between you and coffee. So I'll try and be direct and shorter than I usually am. As we kind of said at the start, we've been trying to do clinical trials in MPS diseases and neuropathic MPS diseases in particular for over the last 20 years. Lots of different trial designs, lots of different endpoints have been used, but we really haven't succeeded as we thought we would. This is a graph from a natural history study of MPS 3A, similar to the graphs that Cara showed actually, and we're repeating this because it's really important to try and illustrate the progression of the disease. The shaded grey line is the development as measured by a standardized test, the Vineland, and the shaded grey area is normal development or typical development. Each of those small black lines is an individual child followed on a natural history study. Again, a child on a study who had repeated assessments, repeated lumbar punctures, and received no therapy whatsoever. And you see, in an attempt to draw the average mean development with the red line, we see from this graph so much about what the challenges are in these clinical trials. So I'll zoom in to the critical bit here. Okay, so time along the bottom, developmental attainment along the top. We see that, as you've seen from many of the videos and the amazing talks from our patient and parent advocates, that these children gain skills normally initially, hence the delay in diagnosis. They have a period of not gaining skills but not losing skills, and then a period of loss of skills or dementia, which I think is a difficult word to use, but actually a very useful word in communicating to people what is actually happening. And so there's lots of different ways we've thought about trying to measure this. As Cara says, of course, we've had this somewhat obsessive approach to neurocognition because it was one of the things that we could measure and put a number on. And those of you involved with trials, we like numbers. We have to have a number for everything. We have to measure everything because without numbers you can't do statistics. So anyway, we've been trying. In the ideal trial for Sanfilippo or a neuropathic MPS, you might get a child while they're developing normally. So that requires the diagnosis before there are a lot of symptoms. So that doesn't usually happen because normally in medicine we make diagnoses because a parent comes to us and complains that things are not correct and there's a problem. So already there's a problem with this ideal trial approach in that we have to get children before they've developed symptoms or before they've fallen off this typical development curve. But if we wait and treat children when they're typically diagnosed at about this point here, when they've already fallen off the curve, this is where the majority of children are. So if you're running a trial in Sanfilippo, the majority of children that you get contacted about are at this kind of stage. So they've already lost skills. And it's very challenging at this point because they're not only losing skills or not gaining skills, they also have this significant behavioral disturbance that you've heard about from so many speakers, which makes it incredibly difficult to administer reproducible, reliable neurocognitive tests. And I think you've heard that most clearly from Kim, and the kids get really fed up with our testing. And so it's uncertain here if we treat here, even if we had a perfect treatment at this point, what is the treatment outcome? What's the treatment outcome at this stage with a perfect treatment? Is it almost can't be returned to normal? Is it a stabilization? Is it a slow improvement? Or is it a slower decline? Almost any of those things could be viewed as a successful treatment. But I think you can see here, if you had stabilization of this child at this point, how many years it would take to follow them before we had clear evidence of a diversion from the natural history of the disease. And in a placebo-controlled trial, this is deeply challenging. So the ideal world, and we have tried to run trials like this in these diseases. We've only accepted in some trials very, very early pre-symptomatic children. It's incredibly difficult to find those patients. Trials have had to have global recruitment. Families have had to move countries, not just cities, to be part of these studies. The vast majority of children have been excluded from these studies. And of course, we can't have newborn screening until we have an effective therapy, or at least a therapy. And so we have to wait. And almost all of the children enrolled onto the trials as very young children, the optimal patient, if you like, as offensive as that is to families, I'm sorry, when we use terms like optimal patient for a clinical trial, they almost always have an affected older sibling, which means that the family who have to move countries, move cities, have to do so with a child who can have very complex care needs and who's not receiving any therapy whatsoever. The challenge, of course, of treating so young is that the time it takes to show a diversion from normal is even longer than when you treat the already affected individual, and maybe 5 to 10 years. And I think the very sobering slide that, Cara, you showed of the number of studies in red that have simply disappeared, and each of those studies was a therapeutic, a drug that may or may not have worked. And I know from looking at many of those studies personally and being part of a number of them, that the majority of those studies were not shelved, disappeared because they didn't work. They disappeared because it was untenable for the company to carry on. That's a lot harder to take. Okay. I want to talk briefly about some studies that I've been involved with, or we in Manchester have, in the UK, have been part of over quite a long period of time now, as you can see, 15 years of clinical trials in MPS 3. And they've taught us a lot, especially about the role, I think, of heparan sulfate. And if we could go back in time and do things better, I think we would. We've already heard from Kim about her son having been on an intrathecal enzyme study. That was for MPS 2, but we took part also in a study, not just for MPS 2, but also for MPS 3A. A very similar trial, again, very early. At that point, we didn't understand CSF heparan sulfate very well. We weren't very good at measuring it, and we probably did things wrong. I'll show a little bit of data. I'll talk about a nutraceutical trial that we ran in Manchester, myself and Brian Bigger, who's in the audience, trying to answer questions about a drug that many families were taking, and Leslie Meltzer from Orchard Therapeutics will talk later on about one of our ex vivo stem cell gene therapy trials. This was an intrathecal enzyme study, okay, for MPS 3A. So intravenous enzyme, as we've heard already, does not cross into the brain, and so the simple approach to deliver enzyme to the central nervous system in this trial was to put a device through that blood-brain barrier and us to simply deliver the enzyme to the spinal fluid itself and hence to the brain. Fritz, who's in the audience, who'll talk later, and I did this study some time ago to great pain. It was very challenging. But the CSF biomarkers at the time were relatively embryonic and overestimated the response. Almost the doses that we use in those clinical trials, 10 milligrams once a month for MPS 2, 45 and 90 milligrams in MPS 3A, once a month again, almost certainly because the CSF heparan sulfate assays used at the time were relatively primitive, that led us to underdose almost all of these patients. And I think that's absolutely fair, as you say, Kim. Despite that, we could see a reduction in CSF heparan sulfate. That was the first time we were able to show that. But it wasn't as dramatic as we initially thought. And so the poor use of a biomarker, the biomarker CSF heparan sulfate, led us to choose an inappropriate dose, possibly an inappropriate dosing interval, and then the reliance on neurocognitive endpoints led to all of these studies for intrathecal enzyme in MPS 2 and MPS 3A failing, despite what was often quite dramatic clinical benefit. And so these studies have simply stopped. And now we know that you can treat central nervous system lysosomal storage diseases with an direct-to-brain enzyme therapy, because the therapy for Batten disease, or CLN2, has indeed been approved in many countries in the world. Interestingly, that drug, Brineura, is given at a dose of 300 milligrams every other week, direct to the ventricles, not to the spine like we were using. I think if we had a better CSF biomarker 15 years ago, we might have done those studies differently, and we might have had a very different outcome. Unfortunately, as our families realize, in rare diseases, we often get one shot at trying to prove whether one of these drugs works or not. And so we have to guess what the right approach is, what the right dose is. And this is really challenging because we only get that one shot, not because there's a rule that we only get one shot, but because there's only so much money and there's only so much time for rare diseases. And that's somewhat unfair, you could argue, for our families. I'm going to show again a trial that we ran in Manchester, an academic study. I'm showing that not because the trial showed clinical benefit. In fact, the trial didn't show clinical benefit. This is a nutraceutical drug, a soya protein, that at this point, when we started the study over 10 years ago, most families with Sanfilippo in the world were already taking, because you could buy it on the internet, based on essentially evidence from fibroblasts, from skin cells. Which was somewhat challenging. So we tried to perform a clinical trial to answer the question. Because there were a number of other studies at the time recruiting really young patients, and because we were just a hospital and a university running the study, not a pharmaceutical company, and all of our funding came from Bob in the UK and the US MPS societies, we were only able to run a 2-year study, and almost all the patients that we were able to recruit were much older, and neurocognitive tests, while we performed them, were somewhat unhelpful and inappropriate as an endpoint. And so we were able to debate and get agreement from the MHRA, the UK drugs regulator, who we'll hear from later on, that in this case we could use CSF heparan sulfate as an endpoint, a primary endpoint for this study, because there was no other option. And if we didn't do the trial, we weren't, there was already risk, okay? So as we've said before, drug approval and trials are all about risk and benefit. There was already risk before we did the trial, because many families in the world were already taking this drug. So if we didn't do the trial, there was still risk and potential harm. So doing the study actually reduced risk, even though we had to accept the non-perfect situation of using a CSF biomarker as our primary endpoint. And in this occasion, it was really useful. We showed that despite all of our effort, despite vast doses of this drug, we were only able to lower CSF heparan sulfate by around 5%. So there was a biological activity, but it was nowhere near enough to have a significant difference, to have a clinically meaningful benefit. And so we could stop. This endpoint allowed us to fail quickly and early, and that was important. There are families don't get many shots at trials. There's only so much money in rare diseases. Failing quickly, because our biomarker told us the answer, was incredibly important because it meant that families, patient organizations, clinicians, scientists can move on and move on to the next, perhaps more likely to benefit therapy. So we showed an example of a trial where CSF heparan sulfate was used, but probably inappropriately, and a poor assay from the dark days. And we've seen an example here of a trial where CSF heparan sulfate was used to really good use and effect. I will finish because I'm going to be in trouble with Sheela. So to summarize, the trial design in these disorders is incredibly challenging. The time course of the disease does not fit our clinical trial model that we so love and use so often for so many different disorders. The perfect study, the pure study, treatment from birth with 10 years follow-up with a placebo group, isn't possible. No one would accept that study, even if anyone could afford it, which they can't. And even if families would enroll themselves into it, which ethically I hope they wouldn't. So we have to find something different. CSF heparan sulfate, I hope we'll show today, can be closely linked to cognitive benefit. There are exceptions to that. So there are, of course, the difference between late treatment and early treatment. But if we are ever to have therapies for neuronopathic MPS diseases, and you'll see some great examples of potential therapies later on in the day, we must approach clinical trials differently. Sorry, Sheela.
Simon, thank you. Thank you so much. And it's, again, as I said, really set the scene for the conversation. It's an opportunity for those of you in the audience or online to ask questions. We have a couple of minutes before the break. Just checking, Sophie, if we got anything online. No questions online. Any questions in the room? If you've used your index cards, Harriet is here to pick them up. You've blinded everybody with science panel and all this brilliant information. Simon, I don't know if you had anything prepared in advance for this session that you wanted to ask as a question to any of our panelists.
I think I may have gone over my time, so I don't know if you wanted to move to coffee.
Okay, well, given that we have no questions, we can give you a few more minutes for coffee. Please, may I ask people to visit the facilities before you get your coffee, because I need you back here at 10:50 to start the next session. Thank you so much. And those of you online, please just turn your cameras off. Do not exit the meeting. It's really difficult to then get you back in. So please turn your cameras off and come back at 10:50. Thank you.